The mosaic part of the fluid mosaic model is made of many components: phospholipids, proteins, and the carbohydrates, which you can see me drawing here. We're mostly going to focus on the proteins. Like all cellular components, they have structures that are suited to their functions. If they're a membrane protein, they have to stay with the membrane. That means being integrated in. We call them integral proteins. Some integral proteins are transmembrane proteins. They cross the membrane and face both the outside and the inside. I've color-coded their polar and nonpolar regions, or at least the polar and nonpolar regions of this transmembrane protein. That often means that pretty much always means that these guys are going to be amphipathic too, just like the phospholipids. They have polar and nonpolar regions so that they float around kind of anchored in those phospholipids. Polar hanging out with the polar, nonpolar hanging out with the nonpolar. Other integral proteins are only exposed on one side of the membrane or the other. The protein on the right is an example of an integral protein that is not transmembrane. So all transmembrane proteins are integral proteins, but not all integral proteins are transmembrane proteins. And there's a third type of protein, the peripheral proteins, that hang out near the membrane but are not integral. They are often associated with the polar part of an integral protein, but since they're on the edge or the periphery of the membrane, they're called peripheral proteins. Transmembrane proteins make up the majority of membrane proteins, and they have a lot of jobs. First, the transport proteins. They're our big focus for an upcoming lesson, and they allow things in and out of the cell. Enzymes conduct chemical reactions. They turn reactions into products. A lot of cellular proteins are enzymes. Some of those enzymes are found integrated into the cell membrane. So this is just one of many places you're going to see proteins acting like enzymes. The next group, I kind of grouped the next two types under the big overarching umbrella of communication. The first is protein receptors, and they're involved in a process called signal transduction, which is a big part of AP Bio. These proteins allow messages to be sent from outside the cell to inside without a substance actually crossing into the cell. These proteins can receive signals from far, far away, nearby, or they can actually interact with a protein that's attached to the membrane of a nearby cell. Either way, they send chemical message from outside to inside the cell. Glycoproteins have carbohydrate tags that allow cells to identify self and non-self. So they're kind of like little ID tags, or like, I don't know, a little lanyard we wear around our neck or something like that. And I grouped the final two types under a joining category. You've already probably learned about gap junctions by the time you're watching this. That's one example of the type of membrane protein that joins cells together. So cells can attach to other cells when their proteins stick to the proteins of other cells. Tight junctions are another example of this. Cells can also attach to the extracellular matrix, which we call the ECM, or the cytoskeleton. So have you ever wondered how our cells can resist gravity, even though we don't have cell walls? Well, in case you did, maybe you didn't, but this is part of it. Cells stay in place and support themselves by attaching their membrane proteins to networks of protein fibers. Those networks outside the cell are called the extracellular matrix. Inside the cell, those protein networks are called the cytoskeleton, and these membrane proteins can attach to either one. They're just proteins sticking to protein fibers. And just to go over a little more about glycoproteins and cell recognition, little branching chains of carbohydrates can be attached to lipids, and we call those glycolipids. Or they can be attached to proteins. We call those glycoproteins. Glycoproteins vary between species, individuals, and even tissue types, so lots of variation. And the most common thing we tend to teach about them in ninth grade is they help identify self versus non-self. So basically your immune system knows what to do with cells, and your body kind of knows where to put things as it's growing and developing. So when you hear of someone needing a transplant match, it likely means that their cellular carbohydrates need to match. So a person that's going to be a good donor has matching glycoproteins on their tissues. However, big however, not all glycoproteins are found in the cell membranes. They are actually, some are floating around as hormones. And I was skimming some recent studies, and they seem to actually be looking at associations between glycoproteins and disorders. So it's not like the protein that's disordered and messed up. It's the carbohydrate on the protein. And we're seeing disorders like cancer or nervous system disorders or metabolic disorders actually linked to the carbohydrates. So we are still learning about these guys. But in short, the carbohydrate chains change the shape of the glycoprotein, and since shape determines a lot of the function, these proteins are likely really important, and there's a lot we can still learn about them. No matter what a membrane protein or membrane proteins do, to be a membrane protein, they have to be made as a protein produced at the ER and transported by the endomembrane system all the way to the cell membrane. So for this little animation or illustration, I'm going to give my phospholipid bilayer two colors, dark blue for one layer and light blue for the other. So I'm changing up how I'm drawing phospholipids now. I'm just going to draw them as like thick lines instead of the individual phospholipids. So we're zooming way out now, looking at a cell rather than like a tiny bit of cell membrane. Since lipids can float within their layer with relative ease and rarely flip to the other layer, you can think of these two layers as working together, but they are able to maintain differences in what they're made of. We actually call this their composition. You'll see used sometimes. The proteins in this illustration are going to be green. So here's a couple that we're going to follow. One has a purple carbohydrate chain so that you can see that the carbohydrates are added on the interior side of the ER. They're put on those proteins and they'll stay with the glycoprotein all the way through. These proteins will move from the ER in a vesicle to the Golgi bodies. I'm going to magically, move them in. And then I'm going to just magically move them to the other side because it takes a lot of time to illustrate this. If you haven't made a flip book in a while, you should try it. Kind of fun, very time consuming. Note that the carbohydrate tag has been facing the organelle's interior the whole time. ER to vesicle to Golgi, always facing the inside or the interior part of that membrane. When a vesicle travels from the Golgi to the cell membrane, the vesicle fuses with the cell membrane, and that also means that the inward-facing proteins and lipids now face outward. It's not important to memorize this. I just wanted you to kind of visualize how it happens in hopes that it helps your understanding of overall like membrane lipids and proteins and how they interact together. All right, that's it for this one. The fluid mosaic, pretty amazing.